Hello, my name's Timothy Paulitz, and I'm a research plant pathologist with the USDA ARS in Pullman, and I'm presently with the Wheat Health, Genetics, and Quality Research Unit. And today I'd like to present a series of videos on soil-borne diseases of wheat and rotation crops that tie in with the book. And I'm going to talk about 4 different fungal diseases and one nematode disease. And what's common about all these diseases is that they're soil-borne, they exist in the soil, and they mainly attack the roots of wheat and other rotation crops. And why are they a problem? Because they rot the seed, they rot the seedlings, they attack the root, and basically they reduce yield and quality. But the problem with a lot of these diseases is they're very difficult to diagnose because they look like a lot of other things above ground. They can be mistaken for drought stress, nutrient deficiency, poorly drained soil, and compact soil. So what I'd like to do in these series of videos is show you what some of the symptoms look like in the field, and then we'll go to some slides to show some other microscopic symptoms and talk a little bit about how they reproduce and grow. And then finally, I'd like to end up with talking about how to manage these diseases. Okay, so Sanford already showed this diagram with all the complex interactions that were going to be involved in crop productivity. And these include biotic factors such as weeds, pests, and pathogens, but also a lot of abiotic factors that can interact. And when we think of crop productivity, we really have to think of all these integrated together. But today I'm really going to focus in on the nematodes and the fungal pathogens specifically. And then I'm going to cover 4 of the fungal diseases, Pythium, Rhizoctonia, strawbreaker foot rot, Fusarium, and then we'll also consider one of the nematode diseases. And then this is also covered in our book chapter 10, where we cover 12 different diseases, but I'm just going to be focusing in on these 5 diseases that are highlighted. So we'll first of all talk about Pythium, which is called the common cold of wheat. This was a term coined by Jim Cook, my predecessor, and what it's really saying is that this disease is present everywhere, present in low levels, and it's sometimes really hard to diagnose unless you have a healthy control to compare. It's used to be classified in the fungi. We now classify it in a different group called the oomycetes, but for all practical purposes, it really exists as a fungus. And the main thing that we'll do is attack the seeds, the seedlings, and the root rot. It's a bigger problem on rotation crops such as peas, lentils, and chickpeas, because that's where we really see the effect. So I'm going to show you a series of videos here. So here we see a field that was infested with Pythium by my colleague Wei-Dong Chen with the grain legume unit. So in this case, all of these fields were planted with the same variety of chickpea, but you can see there are certain areas where nothing came up, and this is because the seed treatment was not effective. So this is the kind of thing you can see if you don't have any protection on the grain legume crops. So to really find out what happens, you have to dig down and take a look at that seed. And here we can see some of that seed that's been pulled up. And you can see some of it has started to germinate, but then has been killed off by the fungal pathogen. So the other thing that you can see, another characteristic of this disease, is if you're looking across a field, you see a lot of areas that are, the stand is much less and very sparse. And this can be another symptom of this disease on chickpeas. So now let's transition over to wheat. So now in the case of wheat, this is also an infested plot that was set up by Kurt Schroeder, my colleague at the University of Idaho. And again, he was also testing out different seed treatments. So the entire field is planted with the same variety, but just has different seed treatments. And here you can see in this clip that one of the plots looks very sparse and lots of open areas in between where the stand has not filled in, and then you can see next to it a much more healthy looking plot. And of course, if you have this very sparse plot, you have a lot of problems with weeds because you don't have the regular competition that can occur. Here's another close-up of 2 plots, the one on the left, healthy plot, and the one on the right that does not have an effective seed treatment. Here we see another one with 3 plots. The one on the left is healthy. The one in the middle is kind of mid-disease, and then that one on the right, you can see that hardly anything came up. And this is a very, very severe form of Pythium on wheat. Again, Pythium on wheat usually doesn't completely kill the seedling, but it just causes stunting. And then here's the close-up of that healthy-looking plot. And then here we see a close-up of the disease plot. So let's take a closer look at some of the important aspects of Pythium. First of all, what are the conditions that are favorable for Pythium? Well, it likes cool, cold, or wet conditions. So this means that it's going to be especially a problem in the spring. It's one of those pathogens that likes high water conditions. And so you're often going to see it in parts of the field that are poorly drained or where you have a tillage pan or something that's restricting the flow. So here's a couple of other field shots again, taken in the early spring. Looking across this hill, you can see a lot of those areas where the wheat has not really filled in. There's another example. And again, you can see that it's kind of in the low parts of the field. So again, the field symptoms on wheat are poor emergence, poor seedling vigor, stunting, and then you'll also see nutrient deficiencies. You'll often see yellowing of the leaves. And a lot of the growers may think that they have a problem with not enough nitrogen, but it's really a problem of this particular disease. And we really didn't know what the impact of this disease was until Jim Cook started to do this work in the 1980s when we had our first Pythium-specific fungicide called metalaxyl. And you can see in this plot here side by side, one plot where he drilled, put metalaxyl in the seed row. The plot on the left is the non-protected check, and you can really see the difference in the stand and the vigor of the plant. So that's where we really got the idea that even though you might not see something as dramatic as you do in chickpeas where nothing comes up, in the case of wheat, we still are taking a big yield hit. And the whole key to this is the fact that Pythium is so fast in getting into those young seedlings. So here's a picture of a seed. You can see that fuzzy white growth around the seed is the hyphae of the fungus. And so this can get into the embryos and the young seedlings very early on, and then for the rest of the season, that seed is stunted. Here's another example of what the roots look like. Remember I said that they were root nibblers. So on the right we have a healthy looking root system. On the left you can see that those roots are quite dramatically reduced. And of course it's not going to be able to take up as much water and nutrients. So here's another example where we actually pasteurize the soil to kill all the Pythium versus a natural soil. So the natural soil is on the right. You can see how fewer roots there are in the test tube, versus on the left with the pasteurized soil, you can see how many more roots there are. So again, emphasizing that this disease is really a root nibbler. And then if we even take a closer look at the roots, this is a microscopic look at root hairs. So root hairs are something that you can't see, but they are responsible for taking up a lot of those water and nutrients. So the picture on the left, we see intact root hairs. Picture on the right, the Pythium has stripped all those root hairs off, and so that will also diminish the ability of the plant to take up water and nutrients. And then finally, this picture shows the reproductive structures that the Pythium forms, these structures called oospores, which can survive in the soil for a long period of time, and then once this root dies, they're then incorporated into the soil. So that's a little bit about kind of the life history. So a few words about control of Pythium. First thing is to avoid planting into cold, wet soils. And so I think all growers know that the soil temperature has to get up enough so that you get good germination of wheat seed. But if you plant into cold soils, a lot of times you can have more of a problem with Pythium. Why is that? Because in cold soils, the plants are slower to emerge, and it gives us a greater window of opportunity for the Pythium to attack. Another thing is to use residue management to speed up soil drying in the spring, and this is especially important in direct seed crops where you have a lot of residue on the surface, so the soil doesn't heat up as quickly. And then also avoid sowing directly after applying herbicides. We'll talk about this later as being part of the green bridge. And then the other thing is maintain good crop nutrition. A lot of growers will use a starter fertilizer when they plant the seed to give the seedling a boost when some of the roots are nibbled away. Second thing is to use fresh seed. Old seed germinates slower and therefore is more susceptible to Pythium. So the recommendation is to use fresh seed from last year, not to use old seed. Now, in terms of genetic resistance, unfortunately, we have no resistance in adapted varieties. We've actually looked for this and done a lot of studies, but it's very rare to find resistance to what we call these necrotrophic pathogens that are nibbling away at the root. Crop rotation, probably not very useful because Pythium species tend to have wide host ranges. So they can attack not only wheat, but all the other rotation crops. So crop rotation is not really effective, although we have found that fallow will reduce the populations, but not completely eliminate it. So that ends up with probably the best way of controlling this disease, and that's with fungicide seed treatments. And of course, most of our growers already apply different things to control smuts and bunts. They often combine them with insecticides, and most of those formulations, if not all of them, are also combined with a chemical called metalaxyl or mefenoxam, and this is very specific for oomycetes. The bottom one I have on there, Thyram's an older product that I don't think is really used much anymore. However, a caveat to this, within the last few years, we've found Pythium isolates that are resistant to mefenoxam. And Weidong Chen has found this to be a problem in chickpeas. And so they're now testing out new chemistries that are effective against this, such as Intego Solo, which is a product called ethoboxin, which is produced by Valent. So that's one thing to keep our eyes out for in the future. Okay, now I'd like to transition to another root disease, very similar to Pythium. We don't have any video on this, but I would like to show you a few slides. It's called Rhizoctonia bear patch in root rot. And there's all sorts of different species of Rhizoctonia, and I'll only focus on 3 of them. But like the Pythium, they have a fairly wide host range. They can attack wheat, they can attack legumes, and they can attack canola. So the 3 groups we'll deal with are Rhizoctonia solani AG8, and this is the one that's most problem on cereals, but can also attack some other crops. And this is the one that causes bear patch. So when you see these pictures, there'll be nothing really that you can mistake this disease for. The second one is Rhizoctonia oryzae, also pathogenic on cereals and some broadleaf crops. And then the last one we'll look at is more of a specific problem on brassicas and canola, and that's Rhizoctonia solani AG2-1. So this picture shows a typical patch that you would see in the field where you have a really acute phase of this disease. So you have these patches that can extend several yards or meters across a field where all the wheat inside that patch is severely stunted. Here's another example of these irregular shaped patches across the field. And then if we take a look at the roots, there's some very specific symptoms to this disease. The first one on the left is what we call spear tipping, where the young seminal roots will all be eaten off and you'll just have the nub of the end of the root. And then the other symptom you can see on that is where the root is pinched off. And this is because the pathogen will attack the outer part of the root, just leaving the vascular system inside. So we sometimes call that pinched root. And then the picture on the right there, you can also see those browning of the root tips, which are very characteristic. Now the main symptom of this disease is stunting. So here we see a picture of healthy and diseased wheat and healthy and diseased barley. And barley is especially susceptible to this stunting characteristic. So it's also rotting the roots, but it's also causing very severe stunting. Now, in a lot of parts of eastern Washington, we don't see that acute bare patch phase, but we see something which I call more of a chronic phase, which is kind of uneven stands. So here we see, looking across some barley fields where the stands are not even, the height is not even. You can see a few spots there where it doesn't look like much of anything came up. Here's another shot looking at barley across a field, and you can see it almost looks like there's dips in and out of the field where the barley is more stunted. Here's another shot, kind of a close-up of one of those areas where the barley has not even closed over completely in the canopy. And then finally, this is more of an aerial shot looking across a field where you can see these holes in the field. And in fact, if you pull up plants from these affected areas and compare them to healthy areas, as I have on the left there, you can see where they're very severely stunted. So that's Rhizoctonia solani. It also, I mentioned it does have a wide host range. We've also found it to attack peas. This is some irrigated peas in the basin where it can cause similar patching in the field. And we've also found the same disease on irrigated onions. So again, the common thing about all those is we see these bare patches in the field. Now the next disease or pathogen that I want to talk about is Rhizoctonia or Rhizae. And the main thing about this is not only does it cause stunting, but it can also kill the seeds off before they come up. So this is an experiment we did in the greenhouse where we're adding increasing levels of inoculum to these tubes. And as you can see, the plants become more and more stunted till we get up to the higher levels, and in fact some of the seedlings or the seeds did not even emerge. So this is typically what we see in wheat. And we've also found it on peas. And in this case, it causes a root rot. You can see the blackening of the roots there, causes a seed rot, and will also move up onto the stem. The final Rhizoctonia we'll look at is called AG2-1. And basically, AG stands for anastomosis groups. It's a way of classifying the different Rhizoctonias. So even though we call them all Rhizoctonia solani, they're really kind of different species. So here we see a picture of canola in the field where it looks like it came up under the straw and then started to die back. And here's a close-up of what it looks like in the greenhouse. And you can see these young seedlings did come up. So it's not the pre-emergence damping off, but then you can see this wire stem or this brown symptom that moves up from the ground and then starts to attack the plant. And eventually that seedling will be killed. So it'll just fall right over and be completely dead. So that's a symptom that we call wire stem. And then out in the field, here's a couple of shots of what I call dead plant carcasses that did not survive the winter next to a healthy plant. And then if you take a look at those and look at the stems, you can see that they have this brown discoloration moving up the stem, essentially girdling the plant and killing the plant. So that pathogen is primarily a problem in canola. Really doesn't do anything to wheat that we can see. Okay, so how do we manage these diseases? And the first thing I want to emphasize is that they tend to be more of a problem in direct seed systems than conventional seed systems, at least when people are just starting direct seed systems. Okay, and part of that is because the tillage breaks up the mycelium in the field, and reduces the ability of the pathogen to move, whereas with a direct seed system, of course, you're not having any tillage. So the three main things we're going to look at are, first of all, green bridge management, and then seed opener disturbance, and then finally seed treatment. So this green bridge concept is one that affects a lot of the different soil-borne pathogens. And it's this idea that they can attack the roots not only of the wheat crop, but also of grassy weeds and crop volunteer. Then when we kill those weeds or volunteer out with herbicides like glyphosate, they can then colonize those dying plants and form a large reservoir of inoculum. And the reason for that is that dying plants don't have their defenses anymore, so these pathogens that attack living plants can also attack dying or dead plants. And so then the idea is if you plant into those dying weeds immediately, you get a large disease loss. And that's why we always recommended for the last 30 years that growers wait between the time they spray out the weeds and when they plant. And just to illustrate this concept, these three slides were all planted at the same time with spring barley. The one on the left, we sprayed out the weeds in the fall with Roundup, and the one in the middle 3 weeks before planting, and then 3 days before planting. And you can really see the difference that has made, how much better it looks where we sprayed out in the fall or 3 weeks before planting compared to the 3 days before planting. Here's another shot of a lot of work we did about 10 years ago with different timings of herbicide spray. So in the background there, the one labeled 4 weeks was planted 4 weeks after spraying. The one in the foreground was planted 2 weeks after spraying, and you can really see how much stunted that looks where we planted 2 days after spraying. And in fact, we did a lot of studies to try to figure out what the best timing was. This graph here just shows that the optimum time is around 10 to 20 days. And if you wait after that, that curve kind of levels off, so you don't get as much of an effect. So that's probably the best way to control this disease. Second thing is something we looked at was seed opener disturbance, especially with direct seeders. You can have two kinds of openers. This first one is a low disturbance disc drill, which causes very little disturbance. And then here we see a hoe drill, which causes more disturbance. And in fact, when we did experiments with this, you can see that we get better stands. The wheat looks better where we have the high disturbance opener versus the disc opener. However, lots of times the yields weren't that different. So we saw an effect early on in the season, but not later. And then finally, seed treatment. Again, all of our growers use seed treatment to control smuts and bunts, and we see some benefit for early seedling growth. Here's an example of a treatment where we just treated the seed with Cruiser, which is an insecticide, and then in the foreground there we used Sedexane, also called Vibrance, a new compound, and you can see how much better the stand is. And then one final example here, Dividend Extreme, which has been used for a fair amount of time versus a non-treated control. Okay, so those last two diseases we talked about are primarily root and seed rotters. So the next two diseases we're going to be talking about are diseases that move up onto the lower stem. And the first is called strawbreaker foot rot, which also goes by the name of eye spot. And this is primarily a disease of winter wheat. It's a bigger problem in the higher rainfall areas of eastern Washington, but you can also see it in the intermediate and dry areas. As I mentioned before, there's a number of names that it goes by, strawbreaker foot rot, or eye spot. It's actually caused by two different fungi, Tapecia eulundae and Tapecia aciformis, and now goes by a different name called oculomaculans. It's soil-borne like the others, but it survives on residue from the diseased plants from the previous year. It also produces spores that can splash up onto the stem, and that's where we get the symptom. And the two main symptoms it causes, because it rots the stem, are lodging, where the wheat will fall down, and then whiteheads, where the flow of nutrients will be cut to the top of the head, and you get much less yield. This is a good example of one of the few soil-borne diseases where we do have good genetic resistance that's been incorporated for the last 20 or 25 years. And we'll talk about that when we look at some of the management techniques. So here's some examples out in the field. This is a variety testing site near Lamont. And actually, this is a bit too late to see good symptoms. We should have come out a few weeks earlier when a lot of the was still green. And then here we can see the first symptom of the disease, and that's called whiteheads. And so while the rest of most of the wheat heads will still be green, you will find in the field some of these that have turned brown prematurely. So this is the first symptom to look for. But remember, there are other diseases that can also cause whiteheads. So the Fusarium Crown Rot, which we'll talk about in a minute, and also the cereal cyst nematode and the Cephalosporium stripe can all cause similar symptoms in terms of these whiteheads. So that's why it's important to really go down to the stem and try to diagnose it. So what you can do then is to take the sheaths, pull them up, peel back the outer leaves to try and look at the inner part of it. And so here you can see where you start to peel them back. Most of these look fairly healthy. Look like a white straw colored. But then you find something that looks like this, which is a typical eye spot symptom. So the main characteristic of this is that it's a very distinct lesion, has a very distinct margin, and it's very elongated. And so this is what you're looking for in terms of the eye spot symptoms. Okay, here's another example of a healthy wheat stem on the bottom and a diseased one on the top, and you can see it looks much more brown in color. And here's another example of a fairly severe infection, and you can see that eye spot on the base of the stem there. Now, the other symptom that's very evident with disease is lodging. And of course, there's lots of things that can cause lodging, such as wind, too high a yield, the heads fall over. But if you see this wheat falling over, this is a good indication it might be strawbreaker foot rot. And you especially see these in these variety testing sites where things have different levels of susceptibility. So here we see a plot where the wheat is completely fallen over. And then taking a closer look, you can see that the wheat has fallen in all directions. And that's another difference between just regular wind blow, which may all be in one direction, whereas here you can see that the weed is falling in a lot of different directions. So let's take a closer look at some specific symptoms. Again, these slides show much more clear, maybe in those videos, what this symptom looks like on the stem. And this is where, again, it gets the name eye spot, because these look like little eyes on the stem. Can be visible when the stems are still green, or visible when the stems have started to mature. So here's a shot of just to demonstrate the effect of variety resistance. So this is a variety testing site. The one labeled Madsen is resistant to strawbreaker. In fact, it was one of the first ones that was derived about 30 years ago. Next to that, we see Eltan, which is a susceptible variety. You can see that plot is completely fallen over. There's one other disease that we don't see very much, but you could mistake this for, and that one's called sharp eye spot, or Rhizoctonia Realice. And if you look at the symptoms of this disease, the margins tend to be much more sharp and distinctive. This is a disease we don't see very often, except maybe early on in the spring on winter wheat, but this is another one that could be mistaken for eye spot. This is really not much of a problem here in Washington. It's a bigger problem in Europe and some other areas that get a lot more rainfall. So how to control this disease? Well, first of all, as I mentioned, there is genetic resistance. There's a number of resistant varieties. You can go to the Washington State Crop Improvement website or the Seed Buyer's Guide, and it will list the resistance of these varieties. So this slide here just shows you some of the current resistant varieties that are out there. You can see a number of ARS varieties. A lot of those are club wheats. Madsen, as I mentioned before, was the original resistant variety. And then a mixture of Oregon varieties, Washington varieties, and even some private varieties such as Westbred. So again, there's good genetic resistance out there. Another thing that can be done is to avoid early planting, and then crop rotation can reduce the residue where the fungus survives. Now, fungicides are also effective if they're sprayed in the spring before stem elongation, and we still have 3 fungicides that are registered, Tilt, Topsin, and Quilt. So that's a possibility of controlling it on susceptible varieties. But since these resistant varieties have come out the last 25 years or so, the amount of fungicides used to control this have really dropped off. So resistance is the most cost-effective way of controlling this disease. Okay, now I'd like to talk about our second crown rot or foot rot, which sometimes people mistake them for, and this one's called Fusarium Dryland foot rot, or also crown rot. And I like to call it Fusarium Crown Rot simply so that growers don't get mixed up with Dryland foot rot and strawbreaker foot rot. So this is a disease caused by actually 2 different species of Fusarium, Fusarium pseudograminearum and Fusarium culmorum. We've done a lot of studies over the years. We find both of them are very, very widespread. We tend to find the pseudograminearum in the hotter, drier areas, culmorum in the cooler areas, but we can find fields where they're both mixed together. And again, the main symptom, like the strawbreaker foot rot, is going to be at the base of the stem. So here we see this brown, chocolate brown discoloration, but the difference is you don't see any distinct lesion. It just looks like a uniform brown coloration. And here we can see another shot. The one on the left shows the diseased ones on the left, healthy, white-looking stems on the right. And then on the picture on the right, you can see a very severe infection where it looks like it's attacking the subcrown internode. And in fact, if it's a very severe infestation, it will actually kill seedlings off. So it can cause a seedling rot in some cases, but that's pretty rare. Like the strawbreaker foot rot, the other symptom we see is whiteheads. And so here we see in a field, and you can see that yellow arrow pointing to a whitehead in the field. So most of the heads will still be green, and then you'll find these whiteheads out in the field. And the big problem with this is because it affects grain quality. So not only yield, but also quality. And it causes smaller seed sizes and lower test weight. So here we see the healthy one on the left where we have US number 1s, 60 pounds per bushel. You can see the one on the right there from a Fusarium infected field. with only 51 pounds per bushel, so a much smaller seed size. And again, that's because the pathogen is reducing that flow of water and nutrients up to the head. Main thing to remember about this is it's favored by drought or water stress. So you can have an infection which really doesn't cause much damage until the plant goes into water stress. It's also increased by excessive nitrogen fertilization. So why are those two things linked? Well, because if you over-fertilize winter wheat, you'll get a lot of vegetative growth over the winter and into the early spring, but then it will quickly deplete the water in the soil profile. Then the plants become stressed, and then the problem becomes more severe. So Jim Cook and Bob Papendick back in the 70s did a lot of classic research showing how nitrogen fertilization can really increase this disease, especially if you over-fertilize. In terms of other things that you can do about management, we don't have any resistance. We've worked on this for a number of years. Australians have developed some what I would call moderately tolerant varieties, but we really don't have that here. Seed treatments have limited efficacy. I have seen some reports out there, but we've never really seen them to be that effective. And so the main control then is cultural practices to avoid early planting and excessive nitrogen. Okay, the final disease that we'll look at is cereal cyst nematode. So those first four that I looked at were fungi in the soil. Now here we're going to look at a nematode, which is a tiny microscopic worm. And so we'll start out with looking at some videos from the field. And one of the symptoms, if it's really severe, is that you have this very patchy looking stand. And so you could say that this is maybe a fungal disease like a bare patch disease, but in fact this is caused by the nematode. And here's another series of shots that look at, rather than bare patches, they look at more uneven stands across the landscape. And so you can see some area there where the wheat is higher, some lower. And so this is from a site that's heavily infected with the cereal cyst. And you can see looking across the landscape that this unevenness of the stand. And then if we take a close look at the roots, and if you look at this one root at the very top that I put a circle around, you can see that the main symptom is what we call like a witch's brooming, or a proliferation of small roots from the site where that nematode has established a feeding site. And we'll see some better pictures in a moment, but that's one thing to look for. And then here's some shots of the female, the white females on the root. They're about the size of a pinhead, and in mid to June to late June, early July, you can pull up plants and see these females protruding from the root. They look like little white pearls. Okay, so now let's look at a few of the slides. Cereal cyst nematode is caused by two species that we have here. Originally, we only thought we had one, and then in 2014, we discovered we also have the Heterodera philipjevi in Washington State as well as Oregon. The picture on the left kind of shows you what these worms look like. They actually have two different life forms. During the early larval stages, they look like those little snakes, like tiny worms. And then when the female advances in life stage, she takes more of a round cyst appearance, which we'll talk about in a minute here. Okay, this picture here shows you a close-up of the nematode mouth part. Now, one of the things I want to say is that nematodes are probably the most diverse and widely numerous invertebrates in the soil. And they form a lot of important ecological functions. So they feed on other fungi, they feed on bacteria, they feed on other invertebrates. So it's really only a small subsample or subset of those nematodes that can attack plants. And the one common thing that they all have is what's called this stylet in their mouth parts. And this is kind of like a hollow hypodermic needle that it can then protrude in and poke into the cell and then suck the cell contents out. So in this respect, it's very much like an aphid. So all the plant parasitic nematodes have this stylet in their mouth parts, which enable them to essentially feed on the plant roots. So here's some examples of what the cereal cyst symptoms look like on the roots. And in all three of these cases, you can see this witch's brooming, this tight proliferation of small roots that protrude from all the places that the nematodes set up a feeding site. And the big problem is that those little stubby roots are not going to be effective in helping the plant take up water and nutrients. And then on the far right here, you can kind of see a close-up there where you can see a feeding site and all these roots protruding. Now here's a close-up of the white females. So the basic life cycle is that the larval stages infect the root, go through 4 larval stages, and eventually the female establishes a feeding site in the root next to the vascular system and then spends the rest of her life there. So she essentially stops moving and will suck up nutrients from that site. And then as she grows, she will protrude from the root, and so you can see her body sticking out here. You can see these white-looking things, which are the young white females. And then eventually she will lay eggs into that, and then her body will then become what we call a cyst, which is a hard outer covering, which will protect those eggs over a long period of time. So then on the right there, you can see all these tiny eggs inside that protective cyst. So once the female dies at the end of the season, the cysts still remain with all those viable eggs. So here's another shot where you can see with the naked eye these white females stuck to the root. So again, this would be something to look for out in the field. And then here's a shot, a close-up of some of those cysts. They start out white, but then as they age, they turn a brown color. And the key thing about those is they can survive in the soil with viable nematodes for many, many years. So this is why it's such a problem, and this is why once it gets established in a field, it's there for many years. And then finally, here's a picture where that cyst is split open, all the eggs are coming out, and then if we take a close-up of the egg, you can see that the first stage larvae is actually developed inside that egg. So you kind of have like a coiled-up snake inside the egg. Okay, let's finish up with talking about some management techniques. First of all, it's very easy to sample soil and have it enumerated. There's a company down in Parma, Idaho called Western Labs that does a lot of nematology sampling. I've given you the website here. You can take soil samples, send it down to them, and they will do counts and identify the cereal cyst nematode. So that's the first thing, determine whether you have a problem. Second thing is that probably our biggest cultural management technique is rotation. So remember I said that the cereal cyst nematode only attacks cereals, will not attack peas, lentils, or chickpeas. So if we plant a non-host crop every 3rd year, that will slow down the development of the nematode because there's basically 1 year where it has nothing to eat. However, it can still survive even that year of not having anything to eat. And that's because those cysts can survive in the soil for many years. The green bridge also applies here, control grassy weeds and to try and break that green bridge carryover. So the cereal cyst can also survive on all of our grassy weeds. Chemical control, there's really nothing out there. There's no nematicides that are registered, and we have not found any effective seed treatments, although some of the companies are working on that. This is very different than a high-value crop like potatoes in the basin, where they do use a lot of nematicides to manage those diseases. So the final and probably the best future solution will be resistance. And we in fact have these resistance genes called these CRE genes, which have been very effective. Australia deployed them about 30 years ago, and cereal cyst nematode is no longer a problem there. So we've been doing a lot of screening of varieties for resistance. And so far, we've come up with a few that are resistant to Philip Jevy, and I've listed some of them here. Syngenta steelhead, ARS Selbu, and Crescent are very resistant. Madsen's moderately resistant. In terms of Aveni, Westbred Rockland is resistant. So we're just starting to get an idea on resistance that's out there. But the other thing we have to keep in mind is resistance is only part of the problem, or part of the solution. We also have to have tolerance, because even in the case of resistance, the nematodes will still get into the root to set up a feeding site. With resistance, they're not successful in establishing a feeding site, and therefore they can't reproduce, so you don't have any new nematodes for the next year, but they can still cause damage. And so that's why we also need varieties that are tolerant to that initial feeding of the nematodes. And so that pretty well finishes up what I'd like to talk about here. And I'm showing this last slide here to talk about our plant pest diagnostic clinic. because a lot of the things I've talked about may be very difficult to figure out, but you can send samples to the Washington State Diagnostic Clinic in Pullman. Rachel Baumberger is the diagnostician. I put some information on here on some of the services that are available and some of her contact information. You can go onto that website and get all the information that you need. So again, defining the problem is always the first part of trying to control it. I've mentioned a number of fungicides in this talk, primarily to control eye spot and a number of seed treatments. Just a reminder that you're legally obligated to follow the label and to only use registered products. I'd like to acknowledge funding over the years from the Washington Grain Commission, from base fundings from the USDA ARS, and of course from this REACH Climate Change Grant, which has funded a lot of this research. I'd also like to thank Dr. Weidong Chen and Dr. Kurt Schroeder at the University of Idaho for their Pythium plots, which I showed earlier. I'd like to thank Kim Campbell, my USDA collaborator, plant breeder, on a number of these projects, especially cereal cyst, and along with her, Dr. Nuan Wen and Yvonne Thompson, and finally, Dr. Bill Schillinger at the Department of Crop and Soil Sciences, who I've collaborated with a lot over the years. Okay, with that, thank you for your attention, and I hope that this has been useful.
One thing that I was curious about, since some of our attendees are coming from the Climate Learning Network side of things,
right.
How does climate affect this? How does dramatic weather affect this? It seems like a lot of those diseases are more prevalent when you have water stress and nutrient stress and things like that. Can you kind of discuss?
Sure. Yeah. In fact, that was one of the big questions when we came into this REACH grant 5 or 6 years ago. And the problem was there really wasn't a lot of good data on how soil-borne pathogens are affected by temperature and moisture. There was more data out there for foliar pathogens. Of course, they're highly dependent on rainfall and on relative humidity, et cetera. So that was one of our first things. And in fact, I had a graduate student that just finished up last July that looked at that question. And we specifically wanted to build a biological baseline to see how they were affected by temperature and moisture. And it turns out that the Fusarium, as we suspected, can grow under very dry conditions, much more so than the Rhizoctonia. But we did a series of experiments to look at that interaction between temperature and moisture in water potential. So that was another confounding factor that we looked at. But the other interesting thing, if we do studies under controlled conditions in the laboratory, and then start to take it to more real conditions where we have other microbes, then a lot of those interactions change. So what you suspect from lab experiments don't actually turn out to be the same. So the bottom line is that the Fusarium, as the name implies, dryland foot rot, dryland will be probably more prevalent under drier conditions. But then if the conditions really get dry and people start to use irrigation, then maybe it's less of a problem. And then the other ones, the Rhizoctonias, the Pythiums, we think those could be a problem under a scenario where you get a lot more rainfall in the springtime, especially for our spring cereals, could exacerbate those. So at this point, we're just able to make kind of generalizations, but more of that work still needs to be done.
Great. We do have a question from the audience. Peter asked, mycorrhiza were used in dealing with cyst nematodes, ? So how are mycorrhiza used in the system?
Yeah, we've never looked at that interaction. In our wheat system here, we really don't have a lot of mycorrhizal infection. And that's probably because we use a lot of phosphorus fertilizers, which tend to inhibit mycorrhizal formation. So really not a lot of evidence on the benefits of mycorrhiza, at least in our wheat system. However, in other horticultural systems, irrigated systems, vegetable crops, mycorrhizae probably do play a more important role. But I've never seen any studies that have looked at interactions between mycorrhizae and at least cereal cyst.
Okay, we have some questions here from Sanford Eigenbrod. He asked, are there other pathogens that we should be alert to now that are spreading in North America?
Yeah, the one that I've got my eye on is something called wheat blast. It turns out it made a species jump in Brazil, become a big problem down there. They've now found it in Bangladesh. It's very similar to a disease called rice blast, which is one of their more severe disease. And so we're keeping an eye out for that. Hope that that doesn't move into the wheat system, but that's about the only one I can see as a potential. Not sure that we have the climate that would foster it, but that's one thing that people are starting to look at. Other than that, it's the usual stripe rust races, stem rust. There was a lot of concern about 10, 15 years ago about new isolates of the stem rust, one called UG99, that started to spread around the world and caused a lot of concern because we didn't have resistance. Turns out, at least in the Pacific Northwest, the stem rust is really the more minor component. We're more concerned with stripe rust, but of course, stripe rust always has new races that are evolving, and the breeders are just trying to keep one step ahead of them.
And further from Dr. Eigenbrod, we had, can you summarize the implications for reduced tillage across the pathogens you've described?
Yeah, so reduced tillage, of all those that I talked about, the biggest implication is with Rhizoctonia. But as I mentioned, it's only during the initial transition that it seems to be a problem. It turns out that once the growers get into long-term direct seeding, we really don't see much of a problem. And that's always stuck in our minds as probably an example where the microflora has adapted to the new conditions. And so the disease becomes suppressed in long-term direct seed. So that's why I tell growers that, it's always a problem during that conversion, but then seems to go away. The other one, the rest of the diseases, it's not as clear. Certainly any disease that's residue-borne, you would suspect would be a bigger problem under direct seed systems because you leave a lot of residue. But I've seen studies that have gone both ways. With the Fusarium Crown Rot, I see it in conventional systems, I see it in direct seed systems. So it's unclear on that one. The rest of them, like I said, the data just seems to go both ways. You've seen some studies where they find them higher under direct seed systems, some lower. The one other disease that we don't have a big problem on is another Fusarium disease called head blight, which is more of a problem in the Midwest and also under irrigated conditions here. And that one is definitely exacerbated by direct seed systems. That's been known for about 20 or 30 years. You leave a lot of residue on the surface. It forms fruiting bodies over the winter. The next spring will produce spores. And so that one's definitely a direct seed disease. But I would say Rhizoctonia is probably the only one of ours that is a big problem under direct seed.
All right, that looks like all the questions we have. So I'd like to thank everyone for attending. As you can see on your screen, this is one of a 6-part series. All of the archives will be available at climatewebinars.net, and the book that everyone keeps referring to is available at that extension.wsu.edu URL that you see on your screen as well. You can actually download a PDF of that book or order one while supplies last, so you can actually have this as a reference covering all these topics. So yeah, next week, if you want to join us, again, climatewebinars.net will be your source for finding that link. It'll work just the way it did this week. You'll log into Zoom about 15 minutes before the session starts, and we will be talking about rotational intensification and diversification. And we hope to see you all there. And with that, I'm signing off as Dan Geller from the Climate Learning Network. Would also like to thank the REACH Project for sponsoring this series, and we'll talk to you next week.